بسم الله الرحمن الرحيم. In this video, we are going to talk about a new subject, which is innate lymphoid cells, and this is the first video about innate lymphoid cells.
This video will be the first one about innate lymphoid cells. Innate lymphoid cells are the innate counterparts of T lymphocytes. They mirror the phenotypes and functions of T cells. The innate lymphoid cells lack the specific adaptive antigen receptors that is generated by recombination of genetic elements, what's called VDJ recombination. So, in contrast to T cells, innate lymphoid cells do not express antigen receptors or undergo clonal selection and expansion when stimulated, as we know in usual adaptive T cells. Natural killer cells can be considered the prototype of the innate lymphoid cells and can be considered also the counterpart of cytotoxic CD8 positive T cells. Also, innate lymphoid cells type 1 and 2 and 3 can represent the innate counterparts of CD4 positive T helper 1 for the innate lymphoid cell type 1 and T helper 2 for the innate lymphoid cells type 2 and T helper 17 for the innate lymphoid cells number 3. Innate lymphoid cells are usually located in mucosal barriers and various tissues, including the intestine, skin, lung, and adipose tissue. However, as an exception, natural killer cells are principally found in the blood circulation, where it constitutes about 5 to 20% of the circulating lymphocytes and are capable of being rapidly recruited to the inflammation sites. The function of these cytokine-producing innate lymphoid cells include appropriate immune responses to commensals and pathogens at the mucosal barriers. Also, they potentiate adaptive immunity, regulated tissue inflammation, and help in metabolic homeostasis and tissue remodeling, and also they make communication or dialogue with the nervous system. In this diagram, we can see the types of innate lymphoid cells. The first type includes natural killer cells and innate lymphoid cells type 1, which are stimulated by intracellular microbes, virus, bacteria, parasites, and by tumors. They secrete interferon gamma, granzyme, and perforins, and they are responsible for type 1 immunity and stimulation of macrophages towards type 1 M1, which is cytotoxic. The second group includes innate lymphoid cells type 2, which are stimulated by large extracellular parasites and allergens, and they secrete interleukin 4, 5, 13, 9, and they are responsible for type 2 immunity and the stimulation of macrophage towards M2. The group 3 include lymphoid tissue inducer, which is stimulated by mesenchymal organizer cells through retinoic acid and other factors, and they secrete lymphotoxin, tumor necrosis factor, interleukin 17, interleukin 22, and rank. They are responsible for formation of secondary lymphoid structures. The fourth group includes innate lymphoid cells type 3, which is stimulated by extracellular microbes, bacteria, and fungi, and they secrete interleukin 22, 17, granulocyte, monocyte, colony stimulating factor, lymphotoxin, and they are responsible for type 3 immunity, phagocytosis, antimicrobial peptides. The LTI, or lymphoid tissue inducer cells, as I explained in the previous diagram, is responsible for formation of secondary lymph nodes and Peyer's patches during embryonic development through the action of lymphotoxin. Lately, another innate lymphoid cells population was described, regulatory population with regulatory phenotype and they are named regulatory innate lymphoid cells. There is no definitive marker universally accepted that can define innate lymphoid cells. That's because their phenotype depends on the tissue they are present in it, and innate lymphoid cells actually represent very heterogeneous populations. Roughly, innate lymphoid cells can be described as CD3 negative lymphocytes that express interleukin-7 receptor. They are CD45 positive, CD3 negative, CD127 positive. However, in many tissues, innate lymphoid cells type 1 do not express CD127. Innate lymphoid cells are responsible for rapid immune response to signals or inducer cytokines expressed by the tissue resident cells. By contrast, the T cell response is slow, takes days, and these cells must undergo clonal expansion to become operational and to develop antigen-specific memory. To remember, there are other types of T cells that are pre-programmed and function like innate cells. These include invariant natural killer T cells, mucosa-associated invariant T cells, diverse subsets of gamma delta T cells, and resident memory T cells. Innate lymphoid cells have major histocompatibility complex type 2, class 2, so they can process antigens, and so they can regulate the activity of antigen-specific T cells. On the other side, T cells produce interleukin-2, promoting innate lymphoid cells activities. So both types of cells generate positive feedback loops that amplify their responses, but they also compete for the same inducer cytokines and survival factors. However, by this way, they ensure timely orchestration of the immune response. As the situation with B cells and T cells, innate lymphoid cells develop from the common lymphoid progenitor, where specific transcription factors will direct the generation of the different types of innate lymphoid cells. Innate lymphoid cells precursors then migrate from their primary site of production into the infected and injured tissues, where they complete their maturation, similar to the differentiation of naive T cells into T helper effectors. In this diagram, we can see the origin of innate lymphoid cells. So we have first common lymphoid progenitor, which will give to common innate lymphoid progenitor, and this one will give rise to natural killer progenitor, which will give rise to natural killer cells, and to common helper innate lymphoid progenitor, and this one will later on give rise to innate lymphoid cells progenitor or lymphoid tissue inducer progenitor. This one, as we suggest, will give rise to lymphoid tissue inducer, and the other one, innate lymphoid common progenitor, will give rise to the three types of innate lymphoid cells, 1 and 2 and 3. We can see here the natural killer cells and innate lymphoid cell type 1 will depend mainly on transcription factor T-bet, you found in both of them. Innate lymphoid cells type 2 will depend on the transcription factor mainly GATA3, while innate lymphoid cell type 3 and a lymphoid tissue inducer will depend on transcription factor retinoic acid orphan receptor gamma T. We will find it in both of them. The nomenclature of innate lymphoid cells was proposed in 2013 when these cells were classified into group 1 and 2 and 3. However, with single cell RNA sequencing, there was identification of more than 50 distinct innate lymphoid cell clusters. However, the subsets within each group have the same output, including cytokines, and their development and function were dependent on the same transcription factors.
I hope you have enjoyed the video, and again I'm reminding you to subscribe to the channel and activating the alarming bell if you are interested in getting any new video, and thank you.